Morning, everyone, and thank you very much for the invitation to present here. I would like to talk about how we use a scanning electron microscope to do ultrastructural histology and immunohistochemistry at a wide scale. And because I know this forum is a good way to reach out to the community and maybe also recruit new young scientists into our field, I was thinking I start my introductory slide with a little bit of background of how I got actually into the EM field. After this, I will talk a little bit how we do electron microscopy at Genentech, and then I will dive into the immunohistochemistry with the scanning EM. Now, my background started as with my undergraduate studies in biology in Germany at the Technical University in Kaiserslautern. And fortunately, already in our undergraduate studies, we had a practical course in morphology electron microscopy. So this was my first dive into the field. However, then, like so many other life scientists, most of the training is then in the more molecular field, and for a while I lost my interest in EM. However, importantly, when I started my PhD in the field of virology, I had two advisors who were very enthusiastic about electron microscopy. And in fact, my advisor Georg wanted to start a collaboration with Jacobine Grinze-Locker at EMBL Heidelberg. And I remember to this day how I asked my advisor, hey, I would love to do this. I would like to learn it. So I was very fortunate that I could take part in the EMBO training course in Immunogold EM, which was at the time organized by Garrett Griffiths. And then I was awarded an EMBO short-time fellowship that actually allowed me to full-time for 3 months nonstop learn Immunogold electron microscopy for the study of virus replication complex. So at the time, we studied the first SARS coronavirus and La Crosse-Bunya virus, both human pathogenic viruses. Now, with this background in Immunogold EM, I applied to several universities in the United States, and I noticed it was definitely a door opener to have a background in Immunogold EM. So I joined the lab of Professor Ann Arvin, who was also an enthusiastic supporter of microscopy for our research. And in Stanford University, I was also fortunate to meet several people who helped me to expand my skill in the EM field. And I would like to especially mention here Lydia Joubert, who was at the time the expert in scanning EM, and John Barino was the expert in transmission EM. And at the same time, I was fortunate that Joanne Buchanan, Stephen Smith, and Christina Micheva actually developed fluorescence array-tomography and conjugated array-tomography, which helped me at developing methods that helped me in my studies of the pathogenesis of the varicella zoster virus. So the training that I got in my postdoctoral research helped me then getting a position at Genentech, where I'm now, and I cannot believe it, already 12 years. Now at Genentech, I'm the EM specialist in the Center of Advanced Light and Electron Microscopy, which is led by our director emeritus. And we are in the Department of Research Pathology. And what makes work in Genentech especially rewarding and interesting is that we are highly collaborative, and we collaborate with many different departments. So for instance, neuroscience, microbiology, immunology. So we are exposed to a very large variety of samples, and this keeps our work interesting because we constantly have to develop new sample preparation and imaging protocols to handle all these different questions. Now, a little bit about Genentech. Genentech is a biotech company located in the San Francisco Bay Area, and you may even know it from the textbook in genetics, because Genentech is really the pioneer of the biotech industry. It made history when it in 1982 produced the first FDA-approved genetically engineered therapeutic medicine. At this time, it was human insulin. And then it made again history by having the first monoclonal antibody therapies against breast cancer with Herceptin and Avastin, and then another monoclonal antibody therapy against multiple sclerosis. So we have currently more than 12,000 employees, and Genentech is now part of the Roche Group. Now at Genentech, we have two major groups that pursue electron microscopy. We have a dedicated cryo-EM group, for structural biology, whose purpose is to elucidate the structure of proteins at very high resolution using mostly single particle averaging methods, and who also recently dived into the field of cryo-electron tomography. Now, I'm located in research pathology, and we are part of the Center for Advanced Light and Electron Microscopy. Our goal is to image all possible samples, mostly with non-cryogenic methods, and this includes a very large field from viruses, bacteria, tissues, and even devices and nanoparticles. Now, our group, the Center for Advanced Light Microscopy, is highly collaborative. As you can see here, the EM part of it is actually very small. So we are a small EM lab. So it's my collaborator, Miriam, and me. However, we are fully embedded in a very tightly knit, highly collaborative network. And this helps us to be really productive. Now, especially what I would like to mention, what helps us is that we have tight collaboration with a necropsy group that always provides us with the best possible tissues that we can get, a histology group who are experts in providing us with the best cryostat sections, an immunohistochemistry group who are experts in developing new antibody protocols and evaluating new antibodies. And then we have access also to experts in multiplexing and in image analysis. Now, because we are a small group, we use two major microscopes, a transmission EM and a scanning EM. And I'm especially in love with our Zeiss Gemini scanning EM, which I will talk about in the next slides. Now, because we collaborate with so many departments, it's important that we have many different sample preparation protocols. And so we specialize on helping our investigators with particle characterization, microbial morphology, high resolution topography SEM, cellular topography SEM, device imaging, and wide scale. And then the wide scale imaging of tissues, which I will talk about in the next slides. Standard immunogold EM, often in combination with negative staining, and correlative EM. Now, when we have a quick look what kind of projects we currently do on either the TEM or the SEM platform, you may be surprised that you will see that there are no tissues analyzed with the transmission EM. Instead, the transmission EM currently is mostly reserved to study at high resolution using negative staining methods. We studied high resolution viruses and cell fractions, or we use it to characterize bacterial morphology and sometimes monolayer of cultured cells. However, mostly of all the tissue questions that we have that come often from safety assessment, we do now all with the scanning EM. And we use backscatter electron scanning EM at a very wide scale. And I will point out in the next slides why this is useful. Now, because we are located in a research pathology department, most of our questions deal with tissues. And if you ever compare the complexity of a monolayer of cultured cell with the complexity of different tissues, you can very quickly be overwhelmed. So it's important to develop methods that can deal with the particular challenges that we have when we image tissues. These are localization. How do you find your needle in the haystack? Then context. How do we make sure we find our way in this complex tissue and don't lose orientation? Unbiased sample. How do we make sure we find the correct area, but we are not biased in our imaging fields? And then most importantly, collaboration. Every party in our collaboration has to understand what we are doing, why we are doing it, and how it relates to their methods. So for instance, a common question is, how does our EM field relate to what has been explored before by light microscopy or standard histology assays? And then, because we are highly collaborative and many of the projects are supported by pathologists, it is important that our data are provided in a way that is open to review and quality control by our collaborating scientists and pathologists. Now, for these reasons, the Backscattered SEM platform is one of the best platforms for wide field tissue imaging. And here are the particular advantages. A scanning EM, in contrast to a transmission EM, has a very large sample chamber. And this allows us to put whole histology glass slides with sections into the SEM chamber. Furthermore, the image is generated by surface scanning, and this makes the workflow very much easier because for many questions where we don't need 3D, where we don't need ribbons for 3D imaging, but we just need very large sections, we can simply create semi-thin or thick sections at a very large size, and we can still get ultrastructural imaging with these. Then an SEM is usually equipped with multiple detectors. This helps in optimizing getting the best images with the best contrast and resolution. And one of the major advantages is because an SEM does not have a CCD camera with fixed field of view, an SEM has flexible scanning fields, and therefore for each question we can optimize the pixel size and the field of view that is most appropriate for the question pursued. Now, here is a quick look of how we mostly prepare our samples in order to optimize this workflow. From the beginning, whenever possible and reasonable, we try to keep our sample blocks as large as possible, in contrast to TM, where you often want to trim them really small. So we usually work with several millimeter by several millimeter large slabs of tissue, and we flat embed them between two Arclar sheets and polymerize them. And then we glue these large slabs onto EPRON blocks, and the EPRON block only serves to clamp it into the microtome so that we can then create parallel section through the whole area of this large sample block. And here you can see how a typical section that we produce compared to a typical TEM section. So we have huge sections with a very large field of view, and importantly, they are not obstructed, so we can find any possible reason and there are no hindrances. Now, we usually prepare our sections on carbon-coated glass slides. In my experience, this has by far been the best platform. We have also experimented with titanium oxide-coated slides and other metal-coated slides, but in my opinion, carbon-coated glass slides are always superior. Now, here's an example how a traditional TM workflow would compare with our new backscattered electron microscopy workflow. In the old times, I had to create many different images with a TEM at a small field and then stitch them together in order to get an overview of a larger area of a tissue, for instance, here an intestinal crypt. The problem is then when you collaborate, you have to show to your collaborator on one hand that you photograph the right area, so you need an overview. But on the other hand, many questions need high magnification. So for instance, here the visualization of desmosomes and tight junctions or mitochondria. Therefore, you need high resolution and have usually a small field of view. Now, in order to keep essentially to understand where these individual images come from, it's often necessary to map them back so that our collaborators understand what we're imaging. And obviously, this also creates a lot of individual images. And if you now also consider that usually we do controlled studies with different dosings, so we may up often with 6, 12, or even more than 30 samples. And in each of these samples, we would photograph several regions with all these images, so you can very quickly get confused. So this is done way easier if you use the large scan field of a scanning electron microscope and you put as many pixels into this field as possible. This way, we can photograph a verified field of view, but we can still get very high pixel resolution. So for instance, this script was photographed with 32,000 by 24,000 pixels, and it allows the investigator to zoom in and then see mitochondria at the highest resolution. Now here's an example of a kidney tissue where we see kidney tubules. This white field was photographed at high resolution, which easily allows to zoom into this field, and then you can see clearly the mitochondria at nice contrast here. Here is a workflow. Here's a typical workflow of how we use this method to support neuroscience projects. So we start with the fixation and harvesting of the brains. And then we process them. As I pointed out, we try to process them so that we retain a large size. We trim, and then usually we produce first T-blue sections. Importantly, these T-blue sections, these wide field T-blue sections, they will be digitally scanned in a bright field scanner, and these images will be shared with our collaborators. Our collaborators have then a chance to point clearly out to us which area is the most important. So what is the region of interest? I take then these digital maps onto my scanning EM and match a parallel section on the scanning EM with these digital maps. This allows me to clearly find what is the region of interest in the SEM section. And then we start photographing first at moderate magnification and then at high magnification in order to get the relevant resolution and field of view. The digital images are then shared either with our pathologists or collaborators, and then we discuss them either qualitatively or, if necessary, they are then quantified digitally. Now, if you find this interesting, if you find this workflow of interest, you can read our manuscript that we published in 2020, where we provide detailed protocols and we provide several examples of the field of ultrastructural pathology and how we use this method for this question. Now, so far, we only talked about morphology SEM imaging. However, many questions really require molecular identification of either organelles, cell types, or antigens of interest or molecules. And for these, you have to essentially be able to develop immunohistochemistry or immunogold protocols on your samples. Now, as you know, this often immunogold and immunohistochemistry at the EM level is very challenging because these are kind of two opposing requirements. On one hand, we want to preserve the ultrastructure as good as possible, and on the other hand, we need to preserve the epitopes. Therefore, you often have to try different methods and screen different antibodies until you are successful. Now, the correlation of the light microscopy or fluorescence microscopy with EM is very useful because the fluorescence microscopy provides a bird-free labeling pattern. This is important because this is the best way to evaluate the specificity of the labeling and screen antibodies. Furthermore, this bird view provides us with the right orientation and it helps us to find a particular structure of interest. And now the EM, on the other hand, provides the better resolution and detailed ultrastructural detail, and our structure of interest can then be studied at higher resolution. Now, at Genentech, we have established two major strategies, and I do not want to go here through all the little details, but what's important is there's one strategy which is known as post-embedding labeling, and the other strategy is pre-embedding labeling. And both have their particular advantages and disadvantages. So mostly we use post-embedding labeling in combination with free substitution in low temperature processing when we have antigens that are fairly highly expressed, so often structural proteins, or that are locally concentrated, like secretory proteins. However, if you're interested in the localization of receptors and lower expressed proteins, these strategy often fail. And for these, we have now developed pre-embedding labeling protocols. Now, importantly, what both of these strategy has in common, I used essentially classic protocols, but I adopted and modified them in a way that they are fully compatible with having this whole workflow on glass slides instead of using any TEM grids in the workflow. And so let me go a little bit through the more details. Now, for our post-embedding labeling strategy, where we use low temperature approaches, we use cryosubstitution, dehydration, and then low temperature processing into low cryo resin. Now, for most of our samples, we do not use a high pressure freezer. And the reason is that the tissues that we get, they're already fixed, lightly fixed, often by perfusion fixation in the animal anyway. So the high pressure freezer it can still provide some benefit, but not as much. And secondly, what we use instead of high pressure freezing, we freeze protect our samples, and then we use freeze substitution. And the advantage of this is that after freeze protection, you can process thicker samples. And this makes it easier to create wide field sections of these samples. Now, we start usually with lightly fixed samples. We freeze protect them in DMSO. We freeze them, and then we usually use freeze substitution in acetone with, and we may add some low amount of uranyl acetate. And a lot of this procedure follows the protocol that Kent McDonald has published in his quick freeze substitution paper. And now we also experimented with different resins. If I'm in a rush, I love to use the K4M resin, which is quickly polymerized and usually provides very good ultrastructure and contrast. But whenever possible, I try to go with the lowest temperature resin, the HM23 resin, because this allows us to do resin infiltration at the lowest possible temperature, with the hope to even better preserve ultrastructure and antigenicity. Now here I would like to show you a real life project where we use this low temperature method in order to address a question at Genentech. And in this case, the company is interested in exploring the use of allele-specific oligonucleotide nanoparticles as therapeutic agents. And a typical project is that an investigator injects these particles into a tissue, and now the question is, where do these ASO nanoparticles actually end up? So the particular question is, which tissue cells and organelles are involved in the accumulation of these nanoparticles? And if you modify these particles, is their route of uptake and their distribution changed? And now in this particular project, we had a pilot study with our Immunohistochemistry lab, and some of their results which suggested that we may have an accumulation in ependymal cells of the choroid plexus. Therefore, we focused on this brain region. Now, we got these brains that were treated with these nanoparticles. And here I just wanted to quickly point out that in this case, for this brain, I used a free substitution cocktail that reduced some glutaraldehyde. And it also even included trace amounts of osmium tetroxide. And I was confident that my antibody still works because in pre-experiment, I had found out that our antibodies are extraordinarily robust. So including some osmium helped me to get later better membrane contrast. Yeah, and so after obtaining our sample blocks, we prepared wide field sections on glass slides, which we could then stain with our antibody against these ASO nanoparticles. And when we then zoomed in by fluorescence microscopy, we could clearly see these very bright spots indicating an accumulation of these nanoparticles, most likely in the endolysosomal system. Now, because we can use a very wide field in the scanning EM, I use this information to guide me of where to look with the scanning EM in this brain region. Now, so like I pointed out, the fluorescence microscopy helped us to find the correct brain region. We use this wide field to find this correct brain region, and now we zoom in the scanning EM until we see these cells in an individual level. And here are two examples of ependymal cells. And when we now focus into these areas here, you can beautifully see that we have here like a lysosome-like structure with lots of 20 nanometer gold particles indicating the localization of these ASO nanoparticles in the lysosome. And I think you can also see that our ultrastructure is very nice. Considering that this is an immunogold labeling protocol. So you can clearly recognize the mitochondria, the ER, and here this endosome-like structure that contains some gold particles, here and here the Golgi apparatus. Now, Like I pointed out, as nice as post-embedding labeling of low temperature treated samples is, it often fails when you have lower expressed molecules, for instance, receptors. And therefore, I saw a need that we definitely had to develop pre-embedding labeling protocols that are compatible with scanning imaging. And here's a quick outline of how we do this. So we start with Cryostat sections that are optimized for electron microscopy, so they are sucrose protected. We place them on glass slides, and then we use very gentle ethanol permeabilization. My preferred labeling protocol always uses a signal amplification step with biotinylated antibodies and then strapped avidine fluoronanogold. And this is wonderful because in the next step, this fluoronanogold allows us to have a quality control and documentation step where we can really see whether our antibody labeled the correct area and has the expected pattern. After the fluorescence is documented, we convert the fluoronanogold particles into larger silver particles. And I highly recommend the Nanoprobe HQ kit because it really works the best. I have compared several. And because we want to work at very wide fields, starting at low magnification, I recommend for many questions to repeat the silver enhancement 3 times for 5 minutes. Then you get nice large gold particles that makes it easier to find this area in the scanning EM. Importantly, because we need some contrast, we do heavy metal staining. However, don't forget that osmium is a strong oxidizer. So if you do silver enhancement, you have to work with low concentration of osmium. Otherwise, you destroy your silver enhanced particles. And then we flat embed these sections, and then we do parallel sections through this original cryostat sections, place them on our carbon-coated grids, and then we can zoom into them with backscattered electron SEM from 50 to often 25,000x as needed. And here are 2 examples. I strongly recommend to first start when you establish these protocols with a very well-characterized antibody. And in this case, we used an anti-KDL antibody, which would label all proteins that have a KDL targeting sequence which locates them to the endoplasmic reticulum. And as you can beautifully see, we have very nice ultrastructure. We have very dense labeling. And the labeling is highly specific. You can see it's really in the lumen of the ER, and you can even see it in the lumen of the nuclear envelope here. So this showed me that our protocol is working pretty well. So I used it then for a more challenging target. And here is an example where we localized the PD-L1 antigen. You may know PD-L1 because it's a very important modulator of the immune response against cancer. And therefore, it's a drug target for cancer immunotherapies. So we thought it would be interesting to develop a protocol where we can localize this protein in different human tissues. And here's an example of a human tonsil tissue. And this is, again, what you see here is a wide field through a tonsil section, which was processed for immunogold EM with silver enhancement. This wide field allows me to find my area of interest. So for instance, here, I wanted to find these different capillaries. Now I zoom into these capillaries, and I see now here several cells, a putative lymphocyte and putative neutrophils. And this wide field also allows me to quickly see whether my gold labeling is specific or whether there's gold all over the place in cells where we would not expect it. Now, by zooming into this neutrophil cells, we could beautifully localize the PDL gold particles here all along the periphery of the cell where you would expect it. And we also know that PDL1 can be internalized, and therefore I'm not surprised that we also see some PDL particles in the inside of the cell. So having this protocol, we use this now to further investigate all positive PD-L1 positive cells and tonsils. And with this, I'm at the end of my talk. I wanted to quickly again really give a big thank you for all the mentors that I had throughout my career, starting with Walter Nagel at my undergraduate studies, my PhD advisors, and then especially Giacomino Grinzelocca, which really got me started in the immunogold EM field. Then my postdoctoral advisors, Carla Kierkegaard and Ann Arvin, and my collaborators in Stanford, Lydia, John, and Joanne. and my first mentors at Genentech, Linda Rengel, who was the EM experts, and Laszlo, who also had an EM background. Then I would like to really thank my colleagues in our Center for Advanced Light Microscopy, my director Meredith, and all my close collaborators. And a special thanks to all our scientists and supporting pathologists for the projects and support. Thank you very much.
Thanks, Mike. This is great talk. There's quite a bit of questions in the chat, and I know we are running out of time, and let's do at least 2 questions, and then we can move the question to the end. And the first question is, I think it's Alice, right? Alice, you want to speak up, or?
I can quickly ask. It's very nice talk, Mike. And one question to me, I know that ICM imaging of the section was really a great adventure for that advantage. So my question is, when you use the glass slides, carbon-coated glass slides, can you use just regular glass slides to coat by yourself by carding, or you have to buy special, very flat commercial glass slides?
Very nice question. No, we prepare all our glass slides ourselves. You can take a standard carbon coater, and I think the setting is about, I would say, 20 nanometers thickness for the carbon. And essentially what you want is a light brown metallic layer of carbon. If it's a too heavy layer of carbon, it may sometimes come off. So our standard glass slides are always the what is it called, superfrost glass slides. So we use superfrost glass slides and carbon coated until it's a metallic light brown level.
Thank you. That's great.
Okay, and the next question is from Tajid. How do you stain your samples? Tajid, you want to elaborate a little bit?
Well, I've actually just looked through the paper, which answered my question. If you want to go ahead and move on to another one.
Okay.
Yeah, maybe, maybe a short comment. So a very important advantage of doing wide field backscatter electron microscopy on glass slide is actually that you really do not need high contrast protocols. So you can literally take an old historic block that was prepared for standard TEM with normal amounts of N-block uranyl acetate and normal amounts of osmium. You do not need any high contrast staining protocol. So we regularly, in fact, we nearly only use standard TEM staining protocols, because I often found when we get too dense metal, this can cause artifacts. And also when you use more traditional TEM staining protocols, I think you may, for some membranes, you may have lower contrast, but overall you have a wider scale of gray levels. And this can also be informative, actually.
Okay, next question from Isabel. What is the thickness of your SEM sections?
Another wonderful advantage of doing wide field imaging on glass slide is that we can accommodate any thickness of interest. So we can image 100 nanometer sections, 500 nanometer sections, up to 1 micro nanometer sections. And another advantage is because you can stain the top of the section. So after we prepare the sections on the glass slide, we do an additional uranyl acetate lead citrate stain. We never get charging, never. It's really solid, robust, no problems.
Okay, we still have a lot of questions. Mike, do you mind to answer from the chat, and so everyone can see your answer? And please stay on at the end. Maybe there we can have some more question time. Great talk.
Okay,
Thanks.
for sure. Thank you very much for the interest.
Okay,